right, good morning. Thank you for attending either in person or virtually. I'd like to introduce our speaker today, Nicholas Fernandez. He is with the Water Institute here at UF as a data scientist. He received his master's and PhD in environmental engineering from the Universidad de los Andes in Colombia. Had a postdoctoral position actually in my home department, soil, water, and ecosystem sciences with Jim Jallis, right? Yeah. So he transitioned from an extensive background in the private sector and environmental consulting, technical sales, and patents to focus on the intersection of data science, water quality, and hydrology. Especially, he works on researching new connections between these fields, harnessing the recent and growing availability of large data sets. He also helps stakeholders to manage their water data, a growing need in times when data-driven decisions are or should be prioritized. With that, you have the floor. Thank you.
Thank you. Thank you, Mark, for the introduction, and I would like to thank also Mr. Kaplan for inviting me and Megan for making this reality. So I titled this talk "Water Quality from Data Scarcity to Abundance," and my background, I work at the intersection of data science, research, and water quality mostly. And thank you for the introduction. I transitioned from the industry. I worked for a long time in several positions, and then at some point I thought to myself, you're not good for you were not meant for this. You're like a scientist, so I decided to go back to school and never left ever since. And then so let's start. I'd like to start the talk showing you a map of Colombia, the country I was born in. I don't know how many of you have looked at it. When I really started paying attention to the map, what first caught my attention was like mostly like half of the country is very empty. It's not many people living there. This is the Amazon basin, and then Orinoco basin, and then one drains to the Pacific. So this part here is quite empty. You see the number of municipalities here, the red dots, is very, very low. And then what also calls my attention later when digging into hydrology is many of the points are above 1,000 meters of elevation, so above 3,000 feet. And that's also that was the normal for me. But if you ask in the US, and even if you search the literature, where do people live? They say in the plainlands or close to a river mouths. But that's not the case in Colombia. So the highest density is at high elevation. And that's probably because we don't have seasons and it's always very hot in the lowlands. So that's probably the reason. So I want to call your attention to the Magdalena Cauca Basin. That's this one right here. About 25% of the area, 80% of the GDP of the country. And what this means, many of us live in the mountains, or used to live, since my family and I now live in the very swampy and flatlands of Florida. And I wanted to thank Universidad de los Andes and my professor Luis Camacho, who had me there in the university for my doctoral studies that I'm going to present. So let's start. This is a map of a small headwater catchment that we had as a case study for my PhD. It's a small catchment, about 200 square kilometers, and it's very agricultural and also overlaps with a lot of coal mining. All those dark squares are coal mining sites, and most of the remaining land use is either conservation or agriculture. And there's a small town, the orange dot on the left, where people live in a more urban area. And it's very high, so above 2,400 meters, so about 8,000 feet and above. And when you go there, this is how the main river looks like: severe eutrophication. For example, there's a lot of pollutants, and probably you see that because there's all activities there. You can see the native plants from Paramos. Those are only found in the very highlands of the Andes and some places I think in Africa they also have paramos. It's a very high mountainous environment. And they're both affected by all activities. So coal mining, you see the plants covered in coal dust, and agriculture, you see just them disappearing. And then it's also easy when you go there, it's also easy to find both industrial and domestic wastewater released to the river without any treatment. And what did we want to do in this catchment? We wanted to evaluate how do we assess water quality when there's scarce information and also highly variable, because you see peaks of concentration. So how do we deal with it? So this is what we did. So this is again a map of the catchment. These are sites of sampling. So and what we did, we had 55 samples, and this is the number I'm gonna call here water scarcity, and I think data scarcity, and I think scarcity comes up to the philosophical question of watching the glass half empty or half full. So, but for this presentation, let's call that scarcity of data. In those 55 samples, we employed cluster analysis to group, see which samples look like each other. And that led us to that classification of colors on the left. And our field visit has assigned a category to each group. So just to make this the story short, so the first cluster is domestic wastewater discharge, and then C2 is the most polluted sites, and then C6 is the main sites on the main river. All other the other groups are different groups, but those are the three most important for today. So that's what the cluster analysis led to. And then we just plotted the groups. So you can see the salt oxygen. So the wastewater, basically anoxic on the left, and on the right, the main river looks very variable from anoxic conditions to saturation. On the group on the right, and that kind of exemplifies what you see for water quality there. And then on the seasonality, we don't have seasons, so I didn't divide on the typical winter, summer. So I divided by the quarters of the year. So the first quarter, the first 3 months, January, February, and March, was the most polluted season of the year. And this is for the salt oxygen. It coincided with other constituents, but this one is a good example. And then those 55 samples had many water constituents included. So how do we find which ones were important? So then we used PCA, principal component analysis, which basically tells us which of those concentrations have a significant variation. And this is what we found on the first components. Basically, many constituents were important, and that's because, as I said, there's many activities, everyone pouring into the river without any treatment, so you get a lot of contamination. So we see pH, bacteria, solids, organic matter, nitrogen, phosphorus, and also on the toxical side, we see many heavy metals as important. And this was like the exploratory analysis in the beginning. And then we said, what do we do with this? So let's try to build a model to make predictions and simulate scenarios. So we decided to focus on the conventional portion of this set. And as you can see, you need to.
Do you mind minimizing the view? So I can see the film. Right.
Can you hold it?
Right.
Okay. So we divided the streams in these three branches. Streams B and C are basically the main river, and then stream A is an uphill creek. And we use a software called Qual2K, developed by Professor Chapra here in the US, because it comprises almost all conventional constituents. And then under data scarcity, when I first came to the US and saw water quality data, you have like very long time series. You have data every day or and there we didn't have that. We just have very sparse observations. So how do you model that? So we use a Lagrangian approach that's basically following a parcel of water going down the river, estimating very well the velocity of the river, and that allows and then collect samples. And if you go with the water, that allows you to estimate the reaction rates of each constituent. And that's what we did here. And we went out 3 times, and we used one data set for calibration, one data set for verification. And this is the example for DO. We did several constituents, I think about 10. And this is the example for DO and organic phosphorus. So we were able to reproduce for both calibration and verification with a different data set, observed data with the estimated rates. And that's what's done over there under scarce data sets. This is the way of doing it. And then with them all, we ask ourselves, let's do some scenarios. So what are the problems and drivers and the importance of headwaters in this region? So the critical scenario, so sometimes when you have a lot of data, you can like plot a distribution of what's the normal condition, the outliers. But if you have only a few records, so we said, how do we imagine a critical scenario? So we defined it as the worst observed concentration. So the highest for most pollutants and the lowest for dissolved oxygen, for example, under typical flows during the polluted season. So we identified the first 3 months of the year as the polluted months. And there's better measurements of flow than concentration. So we did statistics to estimate the flow during those months, and then simulated the worst concentrations, what could happen. And this is the example for bacteria, total coliforms. So right here, in this, the lines, the dotted lines, horizontal lines, are basically water quality standards. And you can see under the critical scenario, we have, this is the main river. Basically, we don't comply with any standard throughout any of the almost 17 kilometers of the river. So we're always more bacteria than allowed. And we do not know how many days a year this is going to happen, but we know it's completely possible because it's been observed. It's been observed. Those concentrations and those flows have been observed. And then what do we need to solve this? So alternative one, well, the obvious response to how do we solve pollution? Let's add a wastewater treatment plant. So this is the first alternative we simulated. So if we add a wastewater treatment plant where the domestic discharge is, what we do is we get rid of this lump over here. And it removes significant amount of load, but then the river will continue to be polluted like 13 kilometers. So maybe the answer is not that obvious. So maybe they need to look into different alternatives. So the second alternative, we asked ourselves, if this is not enough, what should we do? So the only thing that would lower everything to below to comply with regulations would be intervene the headwaters, so the highest points in the catchment. So only after lowering this line here, doing something in the very high lands will lower those amount of bacteria. And then this is the methods we implemented to deal with the scarcity of data and heterogeneity of data in data scarce regions. And then the take-home message is scarce information, and I quote because, as I said, is you can always look at something half empty or half or half full, and that's why I quote the scarce term. Is a powerful driver of better and informed decisions. Here, it highlighted the importance of headwaters and why maybe we shouldn't go for the first intuitive choice for solving the problem. So let's now go to Michigan. Has anyone been in Michigan before? What I love, one of the many things I love about Michigan is local people. They always show you the mini. And they tell you, you're here. You want to go here? You. So, and that's that. Yeah, that's you learn that in like your first week after getting lost. And then a big thank to Michigan State University and Professor Nejad Hashemi, who had me there for my final year of my PhD. And then also wanted to share these pictures that the Red Cedar Creek in Michigan State and the Lake Michigan during the hard winter and the hottest summer. And the reason for showing these pictures is I think I don't really like the oversimplification of the global north, global south, developed or non-developed, but just for the sake of simplicity, if we use those terms, to me, one of the differences between like developed and undeveloped countries is in developed countries, they know and they value that water, regardless of how hot or how cold it is, are places to enjoy, places to have fun, places to fish, to do sports, and then somehow in the South we've kind of forgotten that, and then probably that's where you were before the Clean Water Act, and we still need to drive that pathway, but that's why I wanted to show those pictures. And what did we want to do in Michigan? So they had very tough problems with PFAS. I don't know if you've heard about PFAS. They're also known as forever chemicals, and they're toxic and they do not degrade over time. So they, both local agencies, I think universities and companies were involved. They wanted to start looking at the problem. How do we solve it? How do we start solving it? So they invested time and efforts to go and sample. They identified these sites as confirmed pollution sources, all these. And then they also went out and sampled many wells used as drinking water sources. So it's about 3,000 wells that they went out to visit. And here the number increased, I think 2 orders of magnitude from 55 to 3,000. So a little more information. And this is also rare because in Colombia, usually drinking water always comes from streams. But in in several places in the US, it's mostly wells. And in Michigan is one of those places. So what did we want to do? So we don't know how to find the sources of PFAS or what are the causes. And so we went to state of the art. And we found at that point, there were 4 models that assessed the main drivers of PFAS in different places. So model 1, it was both binary and absolute model. So binary, you want to predict, I either find or not PFAS in a sample. And the absolute, I want to predict how much, what is the concentration in these compounds are usually measured in nanograms per liter. So that's the absolute model. That was the simplest one, only 4 input variables, and then they try to predict in both modes, and it covered the whole continuous United States. For model 2, a little more complex, they had more predictive variables, 21. They had some geology, some hydrology, some soil properties, and it was only a binary model covering the state of New Hampshire, and they harnessed random forest machine learning techniques for doing that. And then the thermal, way more complex, also geospatial, hydrology, and soil properties. But they had a huge database of concentrations of other chemicals. So about 228 different different concentrations. So they had a lot of data. They implemented binary and absolute models in California. And then the last model, also quite complex, about 60 variables, trying to do binary classification in the whole eastern United States. So that's what we found in the state of the art. So when faced with the question, what do we do in Michigan? So let's reconstruct all those 4 models, and we call that baseline models. So what we did was collecting all the variables for all models, collect that information in Michigan, except for the co-contaminants. There were just no database for co-contaminants, so we got those out, but all other, we tried to compile that information as closely as possible. And then we combined the variables. We proposed some additional that we found that could be important in the literature. So overall, about 200 variables, 50 of those proposed. To summarize, some elevation, upstream areas, pH of soil, and we also found some demographics, population and GDP, potentially important. And we did different size of buffers for the variables, 1 kilometer, 3 kilometers, 10 kilometers, to see kind of the size of the impact. And then we employed variable selection techniques to find, we didn't want to make a model with 200 variables. We wanted to make a simple one. So we used Bayesian variable selection to take them down to less than 10 variables. And then we wanted to propose a new approach for modeling. So we ended up doing combined random forest to do the binary classification, detect or non-detect PFAS, and conditional autoregressive model to forecast the concentration. And as I said, now we have data for 8 PFAS species, and we only use 4 to 10 variables as opposed to other more complex models. Now let me show you what we got. So this was the modeling problem. So on the left, the binary problem, we want to either say I will find or not find PFAS. And as you can see, only a few samples find PFAS, and that's because of the detection limits. It's very hard to find PFAS, and it's also very sensitive. And since it's measured in nanograms per liter, it's like very easy to contaminate the sample. It's very sensitive compounds. So that was the classification problem. And then for the detected portion, we wanted to predict the concentration, and this is the distribution of that concentration. So we wanted to be able to predict both. For the PFAS species, this is just the example for the total PFAS concentration. And then for the variables that we found important, we found the proximity of the sources, both the distance and the ID. So which source is nearby was also an important predictor. We found some geophysical properties as important for predicting. So soil drainage class at 1 kilometer buffer, the total recharge at 1 and 3 kilometer buffers, and glacial land systems at 10 kilometers. That's part of the lithology in Michigan. And then for demographics, we found GDP at the county level and population at the census tract level as important predictors for PFAS. So those are the important variables that we found after filtering all those nearly 200. And then with using only those variables, our model turned out to be, this is the first problem, the classification problem. So the new model was as accurate as the other reconstructed models, so above 95% of accuracy. but it was a little, it performed worse for the fine classification of the number of false negatives. It was reduced a little. That's measured with the rock area under the curve metric. And that's perhaps of the many less variables employed, but it was still very accurate for binary classification. And then for concentration, we did two versions of the models. We were better than the other models that did concentration estimation, so better R squared. It's still I think one of the main challenges is still the detection limits. So you only have data for a very few number of the places where you actually detected those concentration, and I think that's one of the main challenges to continue modeling PFAS moving on. So the take-home message: more information, although scarce for detected concentrations, is still a powerful driver for better and informed decisions. Here in Michigan now, they have which are the main drivers, so they can now prioritize monitoring and interventions, thanks to this bigger data set, although sparse in some measurements. Let me now take you to the United States. So this is the continuous United States, and here I think it's 4 orders of magnitude more data. This comes from a recent data set that we put together here at the University of Florida. It's called ChemLotUS, about 35 million of records in about 300,000 sites. And the distribution of the data is only a few sites have more than 1,000 records, and most of the sites have 1 to 10. So in some way, those 1 to 10 sites could also be viewed as data scarce compared to the ones with more information. And again, the pictures, this is a picture from the workshop we had in May with scientists from, I think, 10 universities in the beautiful Ichitope Springs in Florida, just to highlight how water bodies are places to enjoy and have fun. And what we wanted to do here, we didn't want to build a data set. We wanted to, the initial proposal was find the drivers for observed concentrations in big scales. But when faced to that question, we found out it is not, when you download all data available, it is not ready for testing hypotheses. You need to do a lot of work before that. And since there was no ready data set, we spent a lot of time getting it ready. And we found this is a 19-step process that we followed based on best practices to prepare water quality information. It is probably more common for hydrology, like flow or rainfall. Many people knows how to prepare and assess outliers, duplicates for flow data and rainfall data, but for concentration is not as straightforward. So this is the process we followed. It's 19 steps, and some of the biggest one, removing duplicates. There's like 3 types of duplicates I didn't even know of. There's unit conversion is also important. We found several names that could be lumped together because they were similar or highly correlated. And those are the numbers for each of the constituents included in the dataset. So I think it's 11 constituents. And you can see there's a big number of records for all of them. In the end, we got rid the creation process. We got rid of about 1/3 of the original downloaded data. And ultimately, every row was modified, not necessarily the value of concentration, but some metadata about the value was ultimately modified. And then this is how the distribution of the data changed from the raw query to the final data set. So statistical properties change significantly, so that reinforces data when you download it is not ready for making inferences. As for example, here, this is a lump with a different unit that needed to be converted. And then also here, and then it's a long and comprehensive process to prepare the data. And that's the distribution of resulting distribution of the ChemLotUS dataset. We also identified an additional need when you're analyzing water quality. You usually don't analyze only concentration. You usually need to know some more information about the place where you collected the data. What's the area upstream? What's the land use? What's the river width? So there's so many characteristics that you could do like cross analysis. So I saw there's the NHD, the National Hydrography Dataset. The latest version is the high resolution one. And when I saw it, I was just amazed, like how these people spend so much time identifying every river segment in the country and giving it a unique code. When I saw it, I was amazed by that. And then, so what we did was link the water quality sites for every river reach. So, for example, this point here, if this point lies within this polygon, it will be assigned to this river segment, and so on. And that's only possible because of the high resolution of the original dataset. And then, so that's an example of a HOC 12. And so that link is available for all sites in the contiguous United States. And after linking, it's so detailed that most of the river segments are linked to a unique water quality sample. So less than is 95% of the river segments are associated with 3 or less sites. So that helps you refine your analysis. There's still some places that are very densely monitored. One of them is the Tampa Bay. They have, I don't know if it's like every meter. So it's one river segment. This point right here with more than 100 sites is one reach of the Tampa Bay. But we left those sites there because if people want to study the water quality in Tampa Bay, they can zoom in and then decide how they want to disaggregate those data. And then we wanted to give people some examples of how to use this. So this is the concentration of dissolved oxygen by the hour of the day. And then since it's linked to the river reach, we can filter by stream order 3 and stream order 5. So we can see how the higher the order, the higher the concentration of dissolved oxygen. And you can also see the distribution throughout the day. So it goes down when temperature goes up by the middle of the day. We also tested some, what if we compare all headwaters with their corresponding mouths? So we got a statistically significant difference in the pH. So basically mouths are more alkaline than the headwaters. And now when linked to another information, you can start asking more questions like why that happens. And then total suspended solids as a function of the stream order. So solids going up as stream order goes up, except for this one that there's only, in that database, there's only one river labeled as order 11th. I don't remember which one that is, but that's why that doesn't follow the trend. And then we also found the least monitored streams are the first order streams, the headwaters. So even though in the first part of the talk I highlighted their importance, and although they're the most abundant in the US. They're the least monitored. So probably that's something that where you could think there's scarce information in headwaters. So that's probably something that could be improved in the future. So the take-home message, reliable information, either if it's abundant or scarce, science and human will are the main drivers of progress. To me, I added human will to the equation because when looking at this problem, just thinking how much data, like 35 million observations. The other day we were talking at the lab, how much a cheap lab analysis is, and someone said $10, $10 an analyte. So $350,000 for this, plus all the efforts of all the people who went out to the field and collected that, and also the will of the agencies to say, let's put this on a server that everybody can query and download open source. That's only possible with human will, and I find that amazing. And what we're envisioning for this is just finding new connections between the water quality observations, different characteristics, new discoveries, and collaboration with scientists from all over. That's what I have prepared for today. Thank you.
We have time for questions.
Was there, what dry season seasonality in the Colombian data?
The seasonality, oh, there's some seasonality in flow over there. It's, I think in the US is like one mode. Colombia is usually two of flow. I think it's the ICTC. The clouds pass by, I think twice a year. That's why flow increases twice a year. But for water quality, I think it was a combination, because the first 3 months of the year, they're the driest. And if pollution continue, the pollution is the same and it's drier, so less water to dissolve. I think that's why the highest concentrations. I think that's the main driver.
In the example of the Colombian headwater, you mentioned watershed intervention. What specifically were some like the actions that could be taken, and then how did you come up with that amount of reduction initially?
Okay. So first, the amount of reduction, since the model is predictive, you can kind of the reverse problem. How much do I lower the concentration at the start of the model to start being under the regulatory constraints? So that's like an iterative process of lowering down until I get it. So that was just changing the boundary condition. What's the initial concentration I need? And then we did not explore what actions could be taken, but knowing how much I need to lower that, and now people can sit down and say, okay, we need to get rid of this amount of bacteria. How do we do it? So what's the source? Maybe, I don't know, upstream, it's very hilly. So I think it's just regular wastewater getting out of houses, industries. Maybe, I don't know if they have, what's it called, septic tanks. I don't know if they do it, but I think they need to start evaluating what's upstream and start taking measures for that. But we evaluated how much would they need to do and start from there.
You mentioned that not many samples were positive for PFAS in the Michigan study, and that has something to do with the detection limits. So how stringent were those detection limits? Like, could there have been PFAS in samples, but they just did not meet the limits?
Yeah, I think all could have PFAS. I remember the detection limit was for most PFAS species was 2 milligrams per liter, 2 nanograms per liter. So I guess all samples probably had PFAS, because I think it's been proven that pretty much every living organism in the world has PFAS in their blood. So probably all water has, but I think the challenge is how to detect those levels below the two. And I think one of the reviewers highlighted there's some statistical methods that can help you impute the value when there's when it's below the detection level. But it's always going to be like guessing unless the detection limits go down. And I think there was a later paper that highlighted what are the challenges for improve models, improve the detection, because that's otherwise you're never gonna know.
This is a really loaded question, but how do you know when you have enough data? I mean, you said the 55 was too little, and then we look at this, massive data set that you have for the nation, but you got spatial scales here, temporal scales. What do you use as kind of a metric of when you feel satisfied, or when do you think it's like more data than you need? Is there ever such a thing? Not to scientists,
Yeah.
but you're thinking.
I think that's a very open question, and I think if you probably when talking to Matt and Jim and the Water Institute and the Soil Water Ecosystem Sciences Department, I think that was part of one of their proposals and something we want to start looking at. Yeah, what's enough? And at some point there's now a growing evidence that the variability in space is higher than in time. So that kind of tells you if you have time or budget constraints, it's probably better to invest in more sampling sites. But that also depends on your question. But we are working now on examining those trade-offs between more sites or more frequent samples. But yeah, I don't think that's an answer to questions. But it's always I think it's always gonna be a little open and you can always make things better, but it's always gonna be open. How much enough?
Any questions online?
No questions online.
Any more questions in the room? All right, thanks again, Nick. Appreciate it. Never enough sales. Money's the only constraint.
I wanted to ask